And Richard, is your time.
Okay, well, again, thank you very much for inviting me. It's a pleasure to be here. So my name is Richard Rapoza. I am Vice President of R&D at Avid Vascular in California. It's a pleasure to be here again, and we are going to talk about our latest developments in artificial intelligence as it pertains to OCT usage and procedural guidance. So obviously, my conflict of interest is I'm a full-time employee in Avid Vascular, so I couldn't be more conflicted. Thank you. First, some terminology and definitions. Artificial intelligence is a very broad term. It is essentially computer algorithms in general that have characteristics of human thinking. So the algorithm will take various inputs, as many as you want to give it, or as many as you want to program into it, and then make a decision based on those inputs to a more optimal outcome. For example, playing chess based on all the positions of all the pieces, or identifying differences between animals. For example, when you teach it the difference between a cat and a dog, it can tell the difference between a cat and a dog, but it will probably not do too well if you put a goat in the data. So it's all about what you feed it and what comes out. Machine learning is one of the ways of doing it. So in the umbrella of artificial intelligence, machine learning is a technique to teach a computer how to build an algorithm. And it's a popular engineering approach where you use training data. So you have a vast set of data from either a data lake or a specific effort you have endeavored in collecting the data to train the system. And that means constantly comparing the ground truth assessed by somebody who says, this is what the truth is, this is a dog or a cat, and you teach the computer those over and over and over until there's enough data points in the algorithm that you have now trained the algorithm. And then you have to validate it. So you test the inference of that data by applying the trained algorithm to a new data set that you have not used before to confirm that the machine is actually making the right decisions on the new data. And then there's deep learning, which is a subset of machine learning that is a specific computer technique that uses neural networks as a structure for how to structure the information that mimics the human brain. And these neural networks are built out before you train the machine so that all the data gets put in the proper structure in the proper locations and then used appropriately. There's been some key engineering breakthroughs in doing deep learning in the last, I would say, started in the last 10 years, but in the last 5 years specifically, that have led to a lot of AI development in every aspect of society. And in addition, the lower and lower cost of servers and memory banks and computing power has led to also a huge advance in AI. So for example, here you see the Tesla network on autopilot, not released yet, is seeking licenses in many areas in the world, but it's a series of collections of data points. All that is fed into a central neural network that then decides what to do and faces specific sudden pieces of information to make decisions as you go along the way. This technology has advanced quite a bit. In fact, I just heard yesterday in the news that another company, Waymo here in California, is about to get literally about a month away from getting permits to run driverless taxis in San Francisco. So we will have 24/7 taxi service from cars driving around. I see them all the time going across the street here. And they will be commercially launching the service starting in about a month. IBM has long been touting their Watson system, and they've had dramatically different applications for the Watson system, which is a big umbrella branding for all of the AI that they've done, including harvesting information from the internet to be able to answer questions and make mundane conclusions about information, to actually assisting with radiology, for example, in identifying structures in different portions of a scan to assist with the interpretation of those scans. And then more recently, a lot of digital pathology has emerged in order to identify the proper pathologies in the histological sections and assist in interpretation as well. The government has also been trying to keep up and actually not doing badly in terms of applications. And this is a list up to just prior to the pandemic, to 2019 and 20, of approvals that occurred by FDA on different assisted AI-assisted algorithms for the practice of medicine. You can see here pertaining to cardiology, for example, ECG analysis support, cardiac monitoring, automated interpretation of pacing leads, etc. All of those, substantial amount of them approved in different areas, specifically a lot of it in neurology. And you can see the number of approvals that have occurred has really gone up, tamed a little by the pandemic, but then continued as we speak. So we approached this program, this whole thing, using the convolutional network, neural network technique with OCT information, and trained to detect. We trained it to detect pixels in the OCT pullback for both the media and calcium. We prioritized those two primarily because we had invested a substantial amount of money in running the Illumium 4 trial. Many of you participated in the trial. Thank you for that. And in that trial, the premise of the whole thing is that using OCT in addition to angiography will improve your ability to decide what sizing and what preparation techniques you need to use, what sizing of stent you need to use, and to achieve a larger expansion of stents. So the trial has two co-primary endpoints. One is you achieve a larger percent expansion in stents when you use OCT on top of angiography alone, and you also get a better clinical long-term outcome in TLF when you look at 2-year outcomes in the trial. So the two things that we focused on the most, primarily in the beginning, were one, how to properly size a stent by using, when available, the EEL, or the external elastic lamina in the vessel. And then the second thing is understanding where exactly and how big is the calcium, so that you can at least anticipate when you need preparation techniques for the vessel before you put in the stent. And then from there, we could gather a lot of information out of the trial to supplement the algorithms even more. So we, again, proceeded with a convolutional network, neural network trained with pixels, using semantic segmentation, and then highly optimized to process all OCT frames in seconds. So this not only involved the AI effort in and of itself and the algorithm that comes out of that, but also optimizing the way we compute all these things so that it doesn't take 10 seconds, 20 seconds for you to sit there waiting for the answer. It comes up in literally 1 or 2 or 3 seconds to the screen. Otherwise, the user interface and the user experience is not good enough. And so no matter how good the technology is, you can't be sitting there waiting 20 seconds for an answer. Training data obtained from core labs annotated by research fellows was used in all of these algorithms. So we had hundreds of pullbacks from patients in a database, primarily from earlier Illumium trials where the data was stored in core labs. We proceeded to manually annotate half of that data with core lab fellows so that we can create the ground truth and then feed it to the algorithm. And then train the model, develop the model that we deployed was deployed in the Optis Next platform because they have more computer power than the previous platforms. And then we locked the AI algorithms once they were developed, but it's no dynamic learning in the algorithm. So contrary to some popular belief that we are gathering data from pullbacks now with our Ultron system, which includes the AI, we are not. This algorithm is locked. So whatever we trained it with and then validated it with is what it is. There is no more learning. There's no more data collection. If there was data collection, we would have to go through a giant amount of paperwork, specifically even more so in Europe, to be able to get that data into a lake and then be able to analyze it to perfect the algorithm. It's always a possibility in the future, but for now, we have not done it. The system is what it is from the original data. Then we validated that against a separate set of pullbacks and showed that there's greater than 90% sensitivity and specificity to detect EEL and calcium. Those are the first two parameters that we have played with. And here's what the outcome looks like in the Ultrasound system. You can tell where the border is between the outer section of the media and the adventitia from the training. And if indeed you have greater than 180 degrees of detection, we are able to calculate a diameter. If there isn't greater than 180 degrees, we do not display an EEL diameter. We only display a luminal diameter. So in the past, everybody was going by the lumen and then trying to guess what the outer diameter would be if you had to use EEL. The premise in the Illumium 4 trial is that we will, whenever possible, use the EEL to determine stent size. We also use that information here in the longitudinal view, and we display the EEL where possible, where available, greater than 180 degrees in the wall in the white line you see there. So you can more or less detect where healthy vessel is when the EEL is present and the media is not severely thickened. That is intended to be as an aid to pick where the edges of the stent should be relative to the stenosis that you're detecting. And then the calcium itself can be detected, obviously, sometimes not the outer border, but at least the middle and inner borders are easily detected. Calcium has a very unique appearance, so it makes it easier to detect without having to get into more chemical compositions of any kind or anything like that. So we, again, this one is displayed anytime you see any portion of it as an arc. You can adjust in the deeper menus here how much you want to show or not show, depending on what the thicknesses and locations are, so that you don't get distracted with minor portions of calcium. And that is displayed in the angiography screen in the same orange pattern and on the longitudinal view. So we can then make markings on the longitudinal view of where your target area should be, where your MLD measurements are, and those will appear in the angiography later. All of this is gearing towards a procedural assistance type of effort, where we have termed this algorithm for PCI called MLD Max. What we intend to do with that is to draw out the steps necessary to achieve a good result, at least morphologically in PCI, by starting with assessing the morphology and realizing or determining where calcium is and what kind of pre-treatment needs to be applied. Then making the proper length and diameter measurements by identifying healthy landing zones for the length and using EEL for diameter. And then proceeding on after stent deployment to assess if there's any medial dissections, for example, here, any malappositions that could be corrected with post-dilation, and then giving you an expansion of the stent that tries to achieve roughly 80% for acceptable, 90% for optimal. These parameters, of course, the EEL usage here, immediate healthy to healthy length, all those are premises that are built into the Illumium 4 protocol in trial. So Illumium 4 is nearing its last patient follow-up. It is now going to analysis mode, and we intend to publish that trial next year as soon as the data is available. And then that'll hopefully confirm that following this algorithm with OCT on top of the angio guidance will improve results. So the intent here is to use AI to identify a lot of these features. In the future, for example, we could supplement it. We have done calcium here in morphology, and we have done EEL here in diameter. We can supplement it with combining certain variables to calculate an optimal landing zone, for example. And you can use AI for that. And then automated algorithms to detect dissection so that you're not looking at that, so that the system warns you there is a severe dissection, and you can set the level of severity that you like to be warned at. And then that will come up on the screen whenever it happens. And then in the future, we'll talk a little bit about that with expansion, is having all that morphological information in the wall. And knowing the engineering structure of the specific stent you're implanting in terms of material and pattern, you can match the two for a force balance. Obviously, in the end, when you deploy the stent, there's a force balance between the stent pushing and the vessel pushing back. You can develop a model that says, for this brand of stent with this morphology, you're likely to get this % expansion. And if your threshold for expansion is 80%, for example, anything below that would point you towards vessel prepping that needs to happen prior to direct stenting. So that would be an interesting AI development that we are working on currently, to use all of the morphology to be able to predict stent expansion. And at the same time, with the Illumium 4 data, be able to relate that % expansion to outcomes in the future. So let me continue with that. So the software now is structured in this manner as well. We have a section of the software that starts with morphology. It tells you everything that it sees, proceeding on to sizing and telling you what diameters are available and what should be picked for distal and proximal diameters. And then more or less landing zones, not yet automated, but at least visible on the screen. And then a review mode that shows you any information available for these 3 parameters: dissection, acquisition, and expansion. So machine learning for OCT used a lot of training data I mentioned earlier. You can see here the annotation that we did with calcium and the various different features that we annotated to make sure we distinguish between what is calcium, what is not calcium, what's EEL, what's not EEL. Fed all of that greater than 7,000 frames. The beauty here is, of course, I mentioned hundreds of pullbacks that we use, but each frame in those pullbacks counts as a data set in and of itself, because we're only looking at morphology per slice. So that amounted to greater than 7,000 frames to be able to train the algorithm. We use multiple reviews of pixel level accuracy to make sure that we have what we need. And the data review from core labs in the US and Japan was necessary just to cross-correlate interpretation. A lot of OCT is used in Japan. There may have been differences, and it's all about what you feed the algorithm. So we wanted to account for geographical differences in interpretation, or people are oriented towards thinking something is more severe than not, or outlining it a little bit differently. It's all about mimicking that human interpretation. And so we did that both in the US and Japan. Not too much of a difference, but we accounted for both. And then the data was from real-world patients, of course, that all gathered from various Illumium trials, plus additional supplemental data that we got from the core labs. And then all real-world variables were represented in the training as well. So anything around different diseases, percent diabetics, different presentations in patients, all that was taken into account to make the data as inclusive as we could. And that is actually very, very important when it comes to the regulatory assessment and approval of these type of algorithms. Regulatory agencies are very weary that you may have used a very limited data set or a very homogeneous population, and that your results are not extendable to other populations. So we have to prove to them that we have the variety of patients necessary to make this applicable to as many people as possible. Calcium detection and Ultron mentioned earlier was tested on a different a blinded data set after we trained it, blinded from the training. So half of the group that's working on artificial intelligence here in the US is located in Massachusetts, actually. That group, half of it is programming and doing machine training, and then another half of the group is doing validation. And they do not see the original data, nor do they have visibility to the nuances of the algorithms that were developed. All they do is validate what was coming out, and then we reverse the role for the next feature so that people get the intellectual stimulation on both sides. So that proved greater than 90% sensitivity and specificity, as I stated earlier. highly optimized hardware and software pipelines that can process 500 frames in less than 10 seconds. So that's necessary, again, because the user experience will be too delayed otherwise. And I mentioned earlier, the model is locked. There's no additional learning from your own patients. That could be a feature in the future, but not now. Some challenges that we face with this kind of technology. Training data was acquired using Avid Optic system, which is a certain set of hardware configurations, laser wavelength, etc. The catheter itself, the ability of the catheter to reach certain lesions and all that. There's all the physical world limitations on how to gather those pullbacks. The software cannot be used on any OCT machine that is not Avid, because the training was done with Avid catheters and Avid systems. And therefore, it works in a certain way with all those components included. If you use the software with a different pullback in a different machine, it would not be able to work, because not only the platform and the software is different, but the way the data is gathered is different. The machine learning algorithms have the risk of overfitting the data. This is mitigated by ensuring real-world and diverse data training. So we talked about this before, in that if you have a homogeneous set of pullbacks from a specific homogeneous set of patients, you will get the machine to try to predict features in the other patients only that look like the ones in the homogeneous population. So this propagates the problem if you do not make sure that your training data is as varied as possible. Otherwise, the machine will overfit the data and then it will not fit anybody else's data. Sometimes there can be bugs in the AI models, of course, but normally those are really, really hard to identify. The AI model may seem like it's not working right, and usually it's because of its applicability and the training of the original data, not because the model has a computer bug, quote unquote, in it. And that typically needs to be a thing that is clarified with AI algorithms, because people tend to interpret when results are not looking correctly that there is a bug, and there isn't. It's just that the model is unable to predict. If there's a failure, it is not easy to tweak the AI model. You have to pretty much start over. There's nothing in there that you can go back and fix. You'd have to start over and retrain the entire thing to be able to get rid of something specific that you don't like in the behavior of the model. Data augmentation, though, and retraining of model, a continuous training of model, can be used to slowly wash away, if you will, these kind of bugs, but it requires thousands of data points. It's not something that you can go in there and fix and tweak. And then AI needs specialized hardware to run, to run efficiently. And so we have to, this was a big debate internally, because once we develop the algorithms, they take up a lot of memory, and the graphics cards that we need to put on these computers that are in the OCT machines out there are different. So installing the alternate software in older systems requires pretty much a restructuring of the way the computer itself is structured, and changing cards, and hoping that the algorithm runs efficiently for you when it's installed. So, but it is the next level of technology. We have, like I said, taken the approach of using AI to facilitate procedural progress. We have not used it yet for other applications that are more diagnostic in themselves, but we'll talk about those next. So other areas, further procedural assistance development with MLD Max. I talked about this earlier in terms of, for example, coming up with ways to detecting landing zones and optimizing morphologies so that we get the landing zone that's most appropriate, that doesn't lead to an excessively long stent, but at the same time gets the job done in terms of expansion. So there's a lot of assistance there that can be done. Detection of other morphologies, lipid, for example. Lipid plays a huge role in landing zones. You should not be putting stent edges on lipid pools. They are not visible, obviously, in angiography, especially when there's not stenotic, but they are readily visible and very distinct from calcium. It's interesting, though, in contrast to the previous presentation, that of course, if you have chemical information that tells you this is a specific type of liquid that is more dangerous than any general lipid, it will be extremely useful. But in general, since lipid appearance on OCT is fairly uniform, we can tell it very easily from calcium. And calcium and lipid tend to occupy the majority of what you see in terms of morphology in vessel walls. So we are almost done figuring out a lipid algorithm so that we can incorporate it into a landing zone type of procedural assistance. That easily can go into the next bullet, which is thin cap measurements on atheroma. If we can easily detect edges. The resolution of OCT, as you know, is about 20 microns. We have made progress also in the future hardware to use better laser sources and be able to get that accuracy even tighter for measurements. And that'll enable us to be able to measure caps. There's the additional possibility on thin cap atheroma to be able to run some calculations to figure out physically or using tissue characteristics to figure out physically what shapes of atheroma are more prone to burst than others. And then knowing that there's a lipid pool behind and a certain shape of cap and a certain thickness of cap, you can put all that into a model that will predict if the cap is likely to burst or not. The problem being, though, is that of course, in the real world plays out only in part because nobody has run the trial that says these specific things will lead to an event within this time frame. So all of this is almost impossible to relate to real world outcomes unless we take care of the other side of the equation, which is data generation and data lakes that store outcomes in patients based on all of the pullback information that we have will enable us in the future to correlate all of this information and to be able to have predictive models. Plaque erosion is another. It's fairly easy to detect what exactly is on the wall and if it's erosion or not, so that you can avoid stenting when it's unnecessary, and then instead treat medically if in the case of plaque erosion. But of course, that would have to have a trial that proves it prospectively using the pullbacks and then training the model to identify plaque erosion correctly. We have not yet initiated that work, but it's on one of our horizons of activity we need to take on. Thrombus characterization is also possible. It's very possible to see behind in enhanced images because thrombus tends to attenuate quite a bit of light, but we can reconstruct the back if we have minimal information. And then we can also identify the types and shapes of thrombus so we can take inform you of better medical courses of action based on what we see on the pullback. General procedural facilitation using both OCT and angiography is also possible. Just as we can do all of this AI on the OCT pullback itself, we can do it on the angiography as well. For example, we could be looking at angiograms in two angles and telling you what guide wire is best suited to actually navigate that anatomy. We could look at the angiography and combine with the OCT and assist at a more sophisticated level on how we treat bifurcations, for example. That would be very, very useful. We have started doing that algorithm, and portions of that algorithm will be in the next generation of Ultron, coming out towards the end of 23. And then that will have high-level graphics in terms of how to treat the bifurcations, but combining with the at least two angles of angiography will be even more useful. Combining the OCT information with the angiographic information is also possible. Portions of that will be in the next generation of Ultron, where the markings that you see will be immediately placed, and you can zoom in and out of images on the angiography to be able to detect, to tell exactly where the stent should go from the decisions you have made on the longitudinal pullback, now reflected in angiography. Another possibility is to put specific detectable markers on catheters so that you can follow them in angiography and place them where you need to place them exactly, within fractions of a millimeter. So that is all possible using angiography itself and the information that comes from there, and turn that into an AI learning model. Predictive models are also on our horizon. The first one here is stent expansion. You can do a more basic stent expansion model that is more or less related to calcium itself and how calcium expands. We suspect that calcium is 70% of the equation when it comes to expansion. And this would lead to something that, for example, tells you this lesion with a workhorse stent is treatable without any predilation. So direct stenting is fine, you'll get 80% or greater. You can set the percent to whatever you think is good. If you like 75%, you can put it in and it'll tell you this will expand to 75% with X% probability. Something that needs a stronger stent, for example, if there's a product out there that says this stent is a lot stronger and it will work for this lesion, not instead of the workhorse with direct stenting, that's a possibility. And then if we had all other lesions that will show less than X% probability of expanding with any stent, those will require prepping. And then in the future, we would develop models that say, this lesion is more amenable to cutting balloon, for example. This lesion is more amenable to lithotripsy or plain old high pressure dilation. And then based on the plaque morphology, then we could probably detect which one of the techniques will lead to a higher percent expansion probability. Events, like I said, we can start correlating events with Illumium-4. There's not enough data points in Illumium-4, even though we have thousands of patients. There's not enough data there to be able to truly predict events. The models are too broad. We can apply artificial intelligence to the actual clinical data to correlate patient characteristics and OCT pullback characteristics to outcomes in a different way than traditional statistics. But it's still not enough for us to get to the level of accuracy we need to have. So we need to proceed to data lake technique efforts to be able to get enough data for that. And I mentioned earlier, vulnerability of plaque. So those are in the horizon for future developments. We are making slow progress, but we are making good progress in terms of these techniques. Hopefully that will lead to higher utilization of imaging techniques and to a better outcome for patients based on more information that is actually. So I'd be happy to take any questions at this point, and thank you again for inviting me.
Thank you, Richard. Really outstanding presentation. It has been so nice and illustrative to see the two phases, the two aspects of the intravascular imaging. One is really focusing the assistance of the PCI procedure that we are currently using. We are users. And as Richard has shown, we are using the calcium detection. We are using the sizing tools of the CT. We are using Ultron, and it's really useful, really. We are eagerly waiting for the Illumium-4 to see if really this has a translation into a reduction in outcomes. I expect that it's going to be that, it's going to be so. And the other aspect is by Lambros, to see further, to see histology, to make estimation of the future of the plaque, to see beyond the lipid or the calcium, but to see if there is active inflammation. But it's not just currently available, it's more the near future, but it has been very complementary. That's why I enjoyed a lot. Ramiro, Raul, questions for Richard. Richard, the system.
Yeah. Go ahead, Raul, go ahead.
So thank you, Richard, for this. I think we have to be very grateful to Abbott because Abbott is contributing clearly a lot to PCI optimization. And as Chema says, we are using every day OCT for PCI optimization before PCI to decide what to do, and also after PCI to check the stent implantation. And I think it is a matter of time that we have clear data that demonstrate the clinical benefit of using OCT in PCI, at least in complex lesions. My question is beyond the PCI, and I think that now there are some pharmaceutical companies using OCT for their studies on the drugs, on interesting drugs for plaque stabilization to demonstrate that the plaque regression. And this, it is a question that, so as a cardiologist, not as interventional cardiologist, are you seeing more and more companies going to Abbott to use OCT as the way of demonstrating the benefits of some drugs? Are these companies limited to lipid lowering drugs, or are there other drugs such as anti-inflammatory, even genetic? I don't know. We
get typically what happens is that we live in two worlds. Obviously, the device world and the pharma world rarely communicate unless they reside in the same company. But we do get requests for information specifically on who, where, and how is being used, OCT specifically, so that they can pick clinical sites for their drug trials and the subset of sites that will do the imaging that correlates to the treatments. So we collaborate in the sense that if they tell us where to go and who to train, we will go train and then install the equipment and make sure that people know what they're doing when they do the pullbacks. But in terms of the two applications, primarily is lipid stabilization and then from coagulation management. So looking at the specific type of thrombus that is present and then optimizing for that. Those are the two areas that we have seen collaborations more or less form. The one that we have tried to form, which is extremely interesting, and I don't know if you guys saw this publication from the group in ALTS around the PPG index for derived from the FFR pullback or from the pressure pullback in the vessel, correlating the PPG index to angina relief or angina, actually ability to achieve angina relief from PCI. Very interesting because a while back we tried to establish a collaboration with Gilead, a pharmaceutical company here in the Bay Area that looked at anti-anginal usage and optimization of anti-anginal usage based on what we saw in the pullbacks with pressure. And that'll become interesting also when we start looking at microvasculature disease and how to manage that based on either therapeutic techniques or anti-anginals. So it's there's a huge potential for collaboration in all those areas.
Okay, thank you.
Okay, well, I have two very practical questions about the tool, the ultrasound tool. Okay, first of all, how useful do you think is to edit it, the result? Let's say, for example, that I don't agree with the tool that is choosing a strat of the stent and says, okay, this is a strat. So if I say this is a strat, I say this is a malposition of the stent. But I think this is an artifact. It's not a strat really. So what do you think about editing this result so I can correct the conclusion of the tool and I say, okay, this is a good expansion, good or good position of the stent, instead of the conclusion of ultrasound saying, okay, there's a malposition in this segment. You think it could be possible?
It is well. It's possible, of course, to no. We don't tell you what to do, so you can follow your own course of action based on what you see. You can also adjust all of the thresholds for what you are warned about and what you're not warned about. So malpositions that are less than X number of millimeters or X number of percentage expansion, you can adjust all that so it doesn't keep nagging you every time it shows up. But also you can take your own course of action. We don't have a report per se at the end of the case that you have to edit. So at this point, the only thing you can do is ignore it. But there's nothing to change and edit because there's no outcome of that information at the end of the case. It's just stored and it's not displayed if you don't need it to be displayed.
And then the other one is something that I think is a bug of the tool is the algorithm that is used to calculate the percentage of expansion of the stent, because I see many times that the machine says something like 150% of expansion, and it is not correct. It's It's not an exact assumption.
Yes, correct. The algorithm for expansion is specifically that is used in Illumium 4, and is the one that CRF, the core lab in New York, is the one that has published on that algorithm. So it is using the distal and proximal 5 millimeters of area relative to the area in the first ring of the stent. So that's the calculation. It is not the overall expansion, it's not the MLD relative to an average of distal and proximal reference segments. It's purely the expansion of the entry and exit of the stent relative to the immediate vicinity of the stent. We're looking at from Illumium 4, we're trying to then derive what other algorithms are much more predictive of expansion overall than that one, so that we can change the algorithm altogether for the calculation. But that is a pure calculation. It takes a value here, and it calculates a number. We can change that. That's just part of the program. It's not AI, so we can get that done fairly quickly. Once we learn from the Illumium 4 data, what is the actual definition that works the best. So that's still in the works.
Okay, thank you very much. And another question, why do you think, what is the reason to lock the Ultron? So there was no possibility of dynamic learning of the, yeah.
Primarily because it is a huge headache, especially in Europe, to get the permits and the proper authority to be able to collect data from humans. It's a highly protected area in many areas of the world, and data privacy in terms of collecting it and what you do with it. So we did not want to take all that work on. So we said, just keep it where it is. That is a very mundane answer, but it's the truth is that it's a big, huge headache. In the future, though, like I said, we'll never be able to make progress if we don't start a data lake. And we have to, we're already structuring what kind of information we need. So we'll be able to use AI to de-identify information and then store it in a data lake so that we can start predicting outcomes. So it'll be collection of follow-up data, which is required by the European governments, by the way. We have to do post-market data collection on all patients. And so we can use that data to correlate to the original pullbacks and start getting predictive algorithms out there.
Okay, thank you very much. You are muted. You are muted.
Yeah, we are. I want to say that we are running out of time. We could be discussing a lot because we have two exceptional speakers that brought us the cutting edge of the imaging, which is really appreciated. We thank you a lot because we had the opportunity in these two talks and the discussion to see where it's going, what is the next target, what is coming next for intravascular imaging, which is really exciting for us as interventional cardiologists, but also for all the cardiologists and particularly for our patients. No? Thank you, Lambros. Thank you, Richard. We expect eagerly to see you in meetings, in visits to US, or you coming to us. Richard came sometimes to Spain. Lambros, we are also waiting you to come. And thank you to Raul and Ramiro for being discussing and moderating the session. And thank you very much to you both.